So we just understood that our body has different biological characters. But have you wondered what is controlling these biological characters? The answer is proteins. It is different proteins that control different biological characters of our body. There will be different protein that will be controlling our height. There will be different proteins controlling our skin color, hair color, right? So the answer is proteins. Okay, proteins control different characteristics of the body. Now the question comes, how does the cell know? How do these different cells in the body know which protein to produce and how to produce it? Now the answer to that is your DNA. Information about protein synthesis, which protein is to be produced, which amino acids are to be used to synthesize a particular protein, all that information is stored inside your DNA, which is in the nucleus. So however the proteins are made with the information encoded in the DNA by using RNA as an intermediate. Let's understand this statement over here. Soon, our cells faced a problem. What problem did they face? DNA was present inside the nucleus, whereas the site for protein synthesis was our cytoplasm in the ribosomes, right? So the cell faced a problem that the information is someplace else, whereas the site of production of proteins is someplace else. Now how to solve this problem? This problem was solved with the help of an RNA. What our cell does is, it converts the DNA, or rather not converts, copies the DNA to produce an RNA. This RNA is a small molecule as compared to DNA, and even if any damage happens to this RNA, makes no difference to the cell. So this RNA would leave the nucleus, come into the cytoplasm, attach to ribosomes, and now the ribosomes can read information on the RNA and produce proteins. So this process of production of proteins with the information from the DNA using RNA as an intermediate is called as the central dogma, right? Central obviously means rules that apply for all, and dogma means a set of rules. So central dogma are set of rules that are applicable to all. Who are these all that we are talking about? Over here they are all living organisms to which these rules apply. Let's have a look at a few more details. So this is our DNA. It is being converted into the RNA. The RNA is taken into the cytoplasm where it binds to ribosomes. Ribosomes will read information on the RNA. How? We'll see. And according to the information on the RNA, it will bring different amino acids. And as we all know, amino acids when put together form polypeptides, chains of amino acids. And when they are folded in the right manner, they form something called as proteins. So this is how proteins are made, and that is how proteins control our biological characters. Let's go into the depth of this mechanism a bit. So RNA polymerase is an enzyme that binds to the DNA and uses it to produce the mRNA. Very important. We have already studied that there are three types of RNA: mRNA, rRNA and tRNA. So the first one is produced by copying the DNA, called as the mRNA or the messenger RNA. Now we definitely need some enzyme or some chemical to copy the DNA and make an RNA out of it. As you've already seen, the name of the enzyme is RNA polymerase, because it is going to polymerize. Okay, a polymer means a chain. Both DNA and RNAs are chains, long molecules. So it's going to polymerize an RNA molecule with the help of a DNA. Hence the name is RNA polymerase. Only one of the two strands of DNA is used for this purpose. DNA is double stranded. This is something that we already know. But both the strands of the DNA are not going to be used. Only one of the two strands is going to be used. Reason? There are a few differences between DNA and RNA, which we have already studied. Let's have a look at a couple of these differences. First of all, the DNA is double stranded, whereas the RNA is single stranded. That is one basic difference. Second, DNA has a nitrogen base called as thymine. All the other nitrogen bases in RNA is same except for thymine. Instead of thymine, RNA has uracil, and that is why we have different enzyme, which is RNA polymerase, to synthesize this mRNA molecule, right? So the sequence of nucleotides in RNA is complementary to one of the strands of the DNA. Complementary means opposite, right? So if the DNA has A on one end, the RNA is going to be having U, right? I would have said T, but as I just told you, thymine is not present in the RNA. Instead of thymine, uracil is present, right? Now, if on one strand of the DNA, if guanine, G is present, then on RNA what would be present? Exactly, cytosine would be present. So this is called as the complementary sequence of the RNA as compared to the DNA. Alright, so uracil, as we already spoke about it, so this process of the synthesis of mRNA is called as transcription. Now student, this word transcription comes from the word transcript, and transcript literally means a message. Don't you think the DNA is sending a message from the nucleus into the cytoplasm, and that message is in the form of this RNA? Hence it is called as the messenger RNA, and hence the whole process of making this message, making this transcript is called as transcription. A very important process and a very important 2-mark answer for our board exams. Alright, now what happens to the mRNA? How does it leave the nucleus? We already know that the nuclear membrane has a lot of pores inside it called as the nuclear pores. So via one of these pores, the mRNA leaves the nucleus and enters inside the cytoplasm. Once in the cytoplasm, what's going to happen to it? Ribosomes are going to attach to it, and the ribosomes are literally going to read the mRNA like a sentence. Now, if we view mRNA like a sentence, it will definitely be made up of words, right? The only difference over here is each word on this mRNA is made up of 3 letters. In this case, 3 nucleotides. And these words of 3 nucleotides are called as codons. More specifically, triplet codon, because they are made up of 3 nucleotides. Why do we call them codon? Because these are code words. Code words for what? Code words for different amino acids. Now there are 20 different types of amino acids in this entire world. To make the protein that the mRNA is going to give information for, which amino acid should be first, which should be second, which should be third, that information is coded by these triplet codon. Very interesting information over here. So RNA we see, it has so many different codons. Codon number 1 made up of 3 nucleotides, then codon number 2, 3, and an mRNA can have thousands of such codons, right? It was this information we've understood because of the work of Dr. Hargobind Khurana. He actually put in a lot of effort, obviously with the help of his colleagues, to find out which were the codons for different types of amino acids. And this was the table that he gave us, a very important table, which is very important in the recombinant DNA technology, the genetic engineering that we were talking about. And because of that, Dr. Hargobind Khurana was awarded with the Nobel Prize in the year 1968, right? A very important discovery. Anyways, let's move on. So each mRNA is made up of thousands of triplet codons, as I already told you, because the protein is going to be made up of thousands of amino acid, and as each amino acid is coded by a codon, the mRNA itself is made up of thousands of triplet codons. As per the message on the mRNA, the amino acids are supplied by the tRNA. This is where the second RNA comes into picture. Now, inside the cytoplasm, when all the amino acids have been diffused, or when all the amino acids come inside the cell, they immediately attach to another RNA molecule called as the tRNA. Now we call it as the tRNA because it is the transfer RNA. It is this RNA's responsibility to transfer the correct amino acid on the ribosome. When the ribosome will be reading the codons on the mRNA, the correct amino acid is going to be brought by the tRNA, right? So that is what we are understanding over here. So for this process, the tRNA has anticodon. How will the tRNA know, or how will the ribosome know which is the correct tRNA or which is the correct amino acid to be brought? It is because the complementary sequence between the codon of mRNA and anticodon of the tRNA. So the anticodon of the tRNA will enter inside the ribosome and go and bind exactly to the codon of the mRNA. That is how the correct amino acids are brought inside. So what we see in this process, what are the ribosomes doing? It is as if the ribosomes are decoding the coded message of the mRNA. If you remember, the things on the mRNA were called as codons. So the message is in the coded form. What is the ribosome doing over here? Decoding the message. We'll modify the term a bit. Instead of using the term decoding, we'll say the message is being translated. Hence, the name of the process over here is translation. So we have understood two processes so far: transcription, translation. The process of making a message, making a transcript, was transcription, and the process of decoding that message, translating it, is called as translation. A very, very important process. So, the amino acids brought by the tRNA are bonded together by the peptide bonds with the help of rRNA. Now, this is where the third type of RNA comes into picture. For the ribosomes to make the protein, the different amino acids not only need to be brought inside, they need to connect with each other. Now, one amino acid binds with another amino acid with the help of a very strong covalent bond called as the peptide bond. And this peptide bond is made by ribosomal RNA, rRNA, which is, as the name suggests, present inside the ribosomes. Right? So, you can see a ribosome over here right next to me. The thing that it has attached to, the long thread like thing over here, that is the mRNA. There are different codons on it, right? Then you can see there are different sites on the ribosome. Why are these sites? The tRNA is entering. All the tRNAs will try to enter, but only the correct tRNA with the correct anticodon can go and bind to the codon of the mRNA and bring the correct amino acid. Once two tRNAs are inside and once they have brought their respective amino acids, a peptide bond will be created between the two with the help of ribosomal RNA present inside the ribosome. Very, very important process, student. Okay, now moving on. During this process, the ribosome keeps on moving from one end of the mRNA to the other end by the distance of one triplet codon, and this is called as translocation. Obviously, since the mRNA is a long molecule, the ribosome needs to move from one end to another as if it's reading that sentence, right? So, can I say the ribosome is changing its location for reading the mRNA? And that is why this third process is called as translocation. In fact, student, translocation is a part of translation, because in order to translate, translocation has to happen. So, this is the third process, yet another very important process. So, such many chains come together to form complex proteins, not just one. Many of such different complex polypeptide chains could be formed. They could be brought together, attached together, folded in the right manner, and that is how complex proteins are formed. Now, living organisms can produce new individuals like themselves due to genes. This is something that we have already understood. The DNA that is present in our body, we have got from our parents, 50% from the father, 50% from the mother. But every now and then what happens, this process of copying the DNA is not 100% precise. So, every now and then a few mistakes are made. Let's understand about them. Due to this, some of the characters of the parents are transmitted to their offspring, right, as we already spoke about it. However, sometimes certain changes occur in those genes. As I told you, when the DNA is being copied, the copying mechanism is not 100% perfect. Every now and then a few mistakes are made, right. So, sometimes any nucleotide of the gene changes its position. So, they have mentioned us now what is that mistake. The mistake is a wrong nucleotide might get added to the wrong position, right. The sequence of nucleotides might change. This causes a minor change, and this change is called as a mutation. A very, very important driving force for a very important topic that we will be understanding called as evolution.